All right, first I would like to thank the organizers. Good evening, everyone. I am Jiao Sima, a graduate student from David Gilbert Lab. Today I'm going to talk about using deletions, inversions, and transplantations to identify cis-elements that are involved in replication timing control. We all know that in higher eukaryotes, DNA replication proceeds in a temporal spatial order. Clusters of origins fire synchronously during S phase, generating the replication timing program. And a typical example of replication profile is shown here. You can immediately identify domains that replicate early and late along the chromosomes. We know that at least half of the genome changes replication timing during development. And those changes occur at the unit at 400 and to 800 kb levels, which we call replication timing domains. Now, we know that Hi-C divide the nucleus into two distinct self-interacting compartments called A and B. And the open A compartment corresponds to the early replicating DNA, and the closed B compartment corresponds to the late replicating DNA. And those compartments are composed of a smaller structure unit called topologically associated domains, or TAS. Structurally, the replication domain, which are the units of regulation during development, are equivalent to topologically associated domains. So, and we also shown that the topologically associated domains and compartment are established during the early phase of a cell cycle, coincident with the establishment of the replication timing program, which we call timing decision point. So this indicates that replication timing is a functional readout for higher chromatin organization and its properties. So because of the close relationship between replication timing and the 3D organization of the genome, it provides a perfect opportunity to address a central question in the field. To what extent is the structure a cause or a consequence of the function? So in order to answer this question, we want to define cis-elements that will be necessary and or sufficient in replication timing control. In order to do that, we use CRISPRs to generate deletions and inversions to test the necessity and sufficiency of those segments respectively. In addition, we use a bacteriophage system to additionally validate the sufficiency at ectopia loci. So specifically, you want to understand how does the boundary sequences versus the internal DNA segments that contribute to our replication timing, compartmentalization, and organization into TAS or RDs. So the locus we are focusing on is on mouse chromosome 16. It has 3 genes, DPPA4, 2, and MORC. This region replicates uniquely uniquely in embryonic stem cells and switch to late replication upon differentiation to neural precursor cells and other cell types. Hi-C heat map shows that the boundary of this domain aligns with the early side of this timing transition regions consistent with our previous findings. And it's sort of divided into 2 subtypes, in which the first one is an interlat, whereas the other one is lamina bound, as represented by lamin B1 DEM ID. There are a couple of CDCF binding sites within the domain, and there's one single CDCF binding site at the border of the domain. RNA-seq shows that DPPA24 are nicely expressed, and also there is a highly transcribed non-coding transcript very close to the boundary call. P300 marks the active enhancer promoters, and this is the origin as represented by short nascent strand mapping. So the first deletion we generated is deleting this single CDCF binding site at the border. The black is the wild type, red is the deletion, and you can see there's no effect on replication timing upon deletion of this binding site. Next, we broaden the deletion into 45 kb to include this highly transcribed non-coding gene. And as you can see, neither deletion or inversion of this 45 kb has any effect on replication timing. We use the 4C with a bait inside the DPPA domain to see the structural changes upon 45 kb deletion at the boundary. So the black is the wild type, red is the 45 kb deletion. If you were going to overlay them, they're almost identical, suggesting that deleting those 45 kb does not alter test structure as well of this domain. Consistent with our deletion experiments, we found that CDCF is also dispensable for global replication timing program. And it's, as Alfred has already shown, it has minimal effect on the compartmentalization as well. This cell line is actually Alfred's diagram cell line. And we reproduced the result in a CDCF knockdown by siRNA and also knockout. Okay, so next, we generated a series of deletions, start with the same starting point. So the first 335 kb deletion deletes the majority of this timing transition regions, and including the CDCF binding sites close to the boundary. As you can see, there's no effect on replication timing of this DPPA domain upon deletion of those big segments, and withholding the timing transition regions shifted to the left side of the deletion passively. Now, if you include additional 100 kb of the internal DNA sequences, you see a partial delay of the replication timing of the DPPA domain. Okay? Now, further, the 680 kb deletion, which deletes the majority of, but not all, of the DPPA domain, the rest of the DPPA sequence switch to late replication, suggesting that there are multiple elements inside the DPPA domain contribute to the early replication timing of the domain. What's very interesting and surprisingly, with the presence of those 335 kb being present, deletion of the internal DNA sequences at the size of 100 kb or 245 kb did not alter replication timing replication of the DPPA domain, suggesting that those internal DNA sequences works in a context-dependent manner, and they're also redundant. Okay, so because in this cell lines we are comparing homozygous deletions to the wild type, except for this one, we repeated most of the deletions in the hybrid cell line, in which the two alleles can be distinguished by the high density SNPs. Now we screen for heterozygous deletions, leaving the wild type as an internal control. And you can see that the results are highly reproducible. So next, we also had a very interesting inversion of the 680 kb as shown in this blue box. Okay? The half of this region is the TTR, and the right half is the DPPA domain. Okay? Now, if you are going to invert the coordinates, you bring the DPPA internal DNA sequences immediately adjacent to this late replicating gene desert, which are also lamina bound. Okay? And now the TTR, the timing transition regions, goes to the right side. But I want to remind you that the right boundary of this topological of the DPPA domain remain where it is. If you were just going to flip the wild-type replication timing program, it looks like this. And it looks like this for the actual inversion. You can see that the internal DNA sequences of the DPPA domain are sufficient to maintain the early replication timing property without the right boundary. Okay? So to further test the sufficiency of those DNA segments, we adopt this system from bacteriophage. So previously, we have inserted at P elements into a late replicating domain in mouse chromosome 14. We also engineered the at B elements into a DPPA back. Okay? Upon the expression of the integrase, at B binds to the at P and insert the DPPA back in a site-specific manner after recombination. And that DPPA back represents the majority of the interlat region, which are the internal sequences of the DPPA domain. After insertion, they were sufficient to advance the replication timing of the adjacent insertion site. And I would like to let you know that this is about 1.4 megabase long. And we were able to reduce the size of the insertion into 58 KB, removing all the CDCF sites and origins, but leaving only DPPA2 and 4 genes inside it, and still able to reproduce the timing shift results, suggesting that CDCF and origins are dispensable for this activity. And we are now investigating the roles of the genes. So here is a summary of the talk. We found that CDCF is not necessary for timing control of the DPPA locus, and they are not necessary for a genome compartmentalization. Next, we have identified multiple internal segments that contribute to the replication timing of the DPPA domain in a context-dependent and redundant fashion. Okay. So the internal DNA sequences of the DPPA domain are sufficient to maintain their replication property without original TAT boundary. And I want to stress that even very partial sequence of the internal DNA sequence are sufficient to retain the replication property upon insertion of the ectopic site, which really suggesting that the internal DNA sequences play a very dominant role in replication timing control and organization. So with this, I will whisper advice and nice discussion with Peter Fraser and other people. And this is the beautiful coast area of the Gulf of Mexico. And now I'll be happy to take any questions.
So you started by emphasizing how similar replication timing domains and TADs are. Mm-hmm. And how you've cut them to bits and then showed sufficiency of some internal elements for the replication timing. What's happening to the TAD organization?
Yeah, so the 40 experiments are in the pipeline, and we are desperate to know the result.
So is it just about the transcription of the genes?
Okay. First of all, for the very small deletions, we're going to investigate in that. While genome-wide, you can find a lot of exceptions. You can find gene transcriptions in the late regions, and you can also see genes that are not transcribed in the early regions, of course. And we also have inserted the genes into the different location. And even though the selection markers are highly expressed, they are not really driving the replication timing into early compartments. So I think it's in a very context-dependent manner.
But in this particular case, you could just knock out the promoters of the two genes and find out if that...
I haven't I haven't finished knocking out both of them yet, but one of them, when I knock on one of them, nothing happens.
So you might have mentioned this in the beginning, but is this developmentally regulated, this replication timing?
Yes, it is.
So when you transfer this element to a new position, does that confer the developmental regulation of it? Of the timing?
No, this is all, the transplantation is in ESL. So we are not looking at the developmental aspect of it.
Differentiated context. Okay, if there's no further questions,